Bringing chemistry to life, that's the topic, and so I assume they can see the slide, which is great. Good, okay. So I teach first year chemistry. So what I want to do is to bring the molecular world to you. I want to show you a world that you probably haven't seen before. And just to give you an idea of what I've got in mind, you can see on the screen, we're going to be talking about atoms, and atoms with personality. What do we mean when we talk about the personality of an atom? And how can molecules have personality? What are molecules? And then how do we visualize molecules? And then I'm going to talk about the most important substance in the universe. I want you to think about that. Do you think you know which is the most important substance in the universe? And then why is it that carbon-based molecules are so important in living chemical systems? Why does life need carbon? Why is NASA looking for water and carbon out in the universe? Here's a question that might be a bit surprising. How would you like to be remembered after you die? As a diamond or something more useful? I'll give you some chemistry behind that. And then I may not get time to do this because we've got a bit less time than I thought. I was going to talk about how can a chemical system become alive? Because that's what we are, chemical systems. Just chemicals working away and we're alive. And then I'm going to give you one of the biggest ideas that I hope you're going to take away today and really make you think. And it doesn't seem like that sort of thing. Are you like a sand dune? Are you like a sand dune? Okay, so let's start. So very quickly then, what are atoms? I want you to imagine atoms as sort of cloudy balls, okay, like I've got on the screen there at the moment. And the important thing is that they have an electron cloud, negatively charged cloud. The key thing about atoms that you need to know, of course, is that atoms can bond with one another. And so that means they come together and they form a bond by sharing their electron clouds. That's all you need to know. Then if you've done any chemistry at all, you've probably seen the periodic table, which is basically a table which shows all of the atoms available in the universe. So that's basically all of them. There are no other atoms anywhere else in the universe. They're the ones. Now, what do you need to know about atoms? To understand their personality, you need to think about, well, your personality. How greedy are you in terms of money? Like if you walk past someone from the Salvation Army and they're asking for money, do you put money in the box or do you get them to look the other way and take money out of the box? In other words, are you greedy or are you generous? Look at atoms. There's a word for electron greediness. That means how hungry atoms are for electrons. And it's called electronegativity. Can you see the word there on the screen? Electronegativity. And so in the periodic table, the atom with the highest electronegativity, that's the atom that loves to attract electrons most, is fluorine. And the further away you are from fluorine in the periodic table, the less electron greedy you are. It turns out that that's the most important idea that you need to know about atoms, their electronegativity. And so the strongest attraction for electrons are in the top right-hand corner, the weakest attraction for electrons in the lower left-hand corner. Okay. Atoms differ in size, just like you. Some people are big, some people are small, so atoms are large and small. Atoms differ in the number of bonds that they can produce with other atoms. And so this is all part of the personality of an atom. So I won't go into all the detail about this because I'm really just wanting to show you that atoms can then form molecules with different shapes. So what's a molecule? Well, when atoms bond together and form a molecule, molecules have shapes. And so we can show molecules in lots of different ways using a formula, or we can do a ball and stick model, which shows you the shape of the molecule. A space filling model shows you the relative sizes of the atoms when they form a molecule. But you know what? When it all comes down to it, molecules see other molecules as blobs of electron cloud. So that's what I want you to have in your mind at the moment. A molecule is a blob of electron cloud. Now, that electron cloud can be coloured. We can show molecules in different ways. We can use the formula representation, or we can use the ball and stick representation, or the space filling representation. But you know what? At university, we think about molecules as blobs of electron cloud, and we colour them. We colour the cloud according to where the electron cloud is most concentrated. Can you see where it's negative here? So I'll just hold it there. Can you see where the electron cloud is red? So that's where the electron cloud is at its most dense. So imagine a fog. Sometimes the fog is really dense and other times it's not. That's really important because that means that in this particular molecule you're looking at the moment, this end is slightly electron rich, slightly negatively charged, and this end is slightly positively charged. What does that mean? Well, you know that positive and negative attract, yeah? So if you have these molecules together, the negative ends of one molecule would be attracted to the positive ends of molecules around it. So that's all you really need to know now. And so there's this big tug of war in a molecule for where the electron cloud's going to be concentrated. Okay, so how do we visualize molecules? Well, take your running shoes. I don't know whether you're wearing running shoes at the moment, but one of the things that we use in chemistry is to try to explain the properties of substances on the basis of their molecules. And so, you know, the top of your running shoe is actually made up of nylon, and nylon is made up of chains of molecules stacked next to one another and attracted to each other. So it's not very stretchy, but it's really tough. The bottom of your running shoe is made up of rubber. What do the molecules in rubber look like? Well, the molecules in rubber are also long chains, long molecules of chains. And when you squash the chains together, they repel each other and bounce back up again. So what I'm trying to show you here is that molecules have different personalities, and the way you arrange them next to each other, they have different functions. Okay. Can you see atoms and molecules? Well, this is some of the latest research in 2007 where these Japanese workers produced molecules that their models are shown to you here on the left and right here. And there's a technique called high-resolution transmission electron microscopy, and it enables you to see individual molecules. So I don't know whether you can see over here, you can see this little molecule trapped. See that little circle there? That represents the ball end of the molecule, and its little tail flicking away like that. So that's a molecule trapped in a nanotube. So that's an individual molecule. We can actually see molecules, and we can actually put them together like Lego blocks. And in your lifetime, we're going to produce molecular machines. And that's the really exciting area to be in. You can actually design things at the molecular level and become a molecular engineer. So instead of swallowing drugs later on, we'll be swallowing molecular machines. So let me show you some of nature's some of nature's machines. I'm just going to duck out of this for a second. I'm just going to jump a few slides because I'm looking at the time and where we're going. So what I might do is I'll start with something really simple like water. Water is the most important substance in the universe. Without water, you're not going to have life. And that's anywhere in the universe. You need carbon and you need water. So I'm going to take you now down into the molecular world of water. Can I ask you to imagine, what do you think water is like at the molecular level? You know the formula for water is H2O. So a water molecule contains 2 hydrogen atoms and an oxygen atom. So let me take you now on this little journey and zoom down, and I'll show you liquid water, a representation of it.
Let's imagine what liquid water would look like at the molecular level. Lots of activity, but do you notice anything? If you look carefully, some of the water molecules are moving in clusters.
Okay, so with that model of water, can I ask you to now think about ice? I showed you the water molecules in liquid water. How do you think the water molecules in ice are behaving? On the screen there's 4 options there for you. The first one says, oh, the molecules will be moving around, but just much more slowly because ice is at a lower temperature. The second option is that the molecules wouldn't be moving, they're just vibrating in ordered positions in space. The third one says, no, the molecules wouldn't be moving because it's a solid, but the water molecules would be in ordered positions in space. And the fourth option says the molecules would not be moving, but in random positions in space. Okay, come on, commit, get a pen, write down 1, 2, 3 or 4. Come on. What are you going to say? Which one? What do you think? Commit so that people know what you said. You put your whole scientific reputation on your answer to this.
They can call out loud. If they want to unmute, they can answer.
Okay. Oh, okay. So I've got 2. That's great. Okay. All right. Right, so watch the screen and I'll show you an animation of ice and see whether you're right.
Now let's look at ice. How is it different at the molecular level? Notice that the positions of the molecules are fixed in space, but they're still vibrating. They never stop vibrating.
Well done. So if you said 2, you were right. And you'll notice that the water molecules in ice are slightly further apart than they are in liquid water. That's really unusual. Usually the solid state is much more dense than the liquid state. Water is one of the most unusual substances because the solid is less dense than the liquid. Why is that important? That's one of the reasons why water is so essential for life. Because whenever ice forms, it always floats on the top of the water, and therefore living systems can still operate in the liquid water below. So water is actually an incredibly special substance. I haven't got time to talk more about that, but it is really amazing. So let's have a look at living systems and look at some carbon-based molecules. You've all seen bacteria, and a lot of bacteria have little tails that flick round, move around like that. That means that there must be a little molecular motor, there must be a little molecular machine inside that bacterium, which is switching that molecular tail along. So let's zoom down now and have a look inside a bacterium. So here we go. So that's a representation of a slice through a bacterium. Can anyone tell me what they think that long molecule inside the bacterium is, represented in yellow there? A long molecule that's really important in living systems. It determines what the species is.
Richmond has a nucleus.
The nucleus in a bacterium like that, there isn't a nucleus. In fact, there's just really the cell. It's just a bag. But it's a really important molecule. You hear about it all the time, and it's such an important molecule in living systems. And it determines what that species is. DNA? Have you ever heard of DNA? Well, that's DNA. But now what I want to do is see right at the very top. Did someone come up with that?
Richmond.
Ah, Richmond, clever Richmond. Yes, well done. Let's zoom down and have a look at the top there where that little flagella goes in there. And let's take a closer look. So we're peeling away, zooming down. And can you see, this is where because the tail is made up of molecules all slotted together. And this little rotor rotates around really, really fast. That's a molecular machine inside a bacterium. It's really fascinating that the bacteria learnt a long time ago how to do amazing things at the molecular level. And so if we keep on zooming down, there's another molecule there. If we keep on zooming down, see the little dots there? They're the relative size of the water molecules in the bacterial cell. So what I really want to show you here is that a bacterium is just chock full of molecules moving around doing things. And so my next slide, just write this down if you're interested. This is a website. Just type in the URL pdb.org. pdb.org. That's a research level site, but you can go there and you can see models of all of the important molecules that are being produced and that are being identified in research today. And it has an educational side to it too. So it's a really exciting place to delve into the molecular world. Okay, so let me now show you on the screen at the moment, what do you think that is? It's a representation of molecules. Can you recognize what that might be? Can you remember can you recognize what you think we're cutting through here might be? So if you've done biology, you might recognize that. That's a membrane. That's a membrane around a cell. And can you see all these molecules embedded in the membrane? Those molecules give the membrane its identity. See, when cells in your body are squashed up next to each other, and when other cells come up and approach, they recognize each other by the molecules embedded in the membrane. Now I'm going to show you a really interesting visualization. Write this down too if you want to actually get a copy of this animation. It's called the Harvard Cell Movie, or Inner Life. Harvard Cell Movie. It's a beautiful, very accurate representation of what it might be like inside a cell where all the water molecules have been taken out and made invisible. And so on the screen at the moment, what you can see is at the top, you can see the surface of the membrane. So we're inside a cell now. And so there's the membrane at the top. And these molecules here are keeping the membrane from collapsing in. And this molecule here on the left-hand side, this is like the scaffolding in a building. So inside a cell, there are these actin fibers that are a bit like the posts and the scaffolding that stop the cell from collapsing and give it some structure. So watch this, and I'm going to show you in a minute my favorite molecule, because I'm going to show you how molecules get around inside a cell. You might think that they just drift along, but that's far too slow for most cells. Things have to happen quickly. So how do you think molecules get from one part of a cell to another part? Have a watch of this. So we're looking at the top of the membrane. We're looking at the membrane at the moment. And here are the actin fibers. They're like the posts, the scaffolding within the cell. And when a cell changes shape, new scaffolds are produced. And then some of the scaffolding is cut up, broken, and new scaffolds. Now these are the microtubules. They are the roads in a cell. And you'll see what I mean. Look at that. That's a molecule called a motor protein, walking along a microtubule, pulling a big bag of chemicals behind it. And so these molecules are amazing. They're called motor proteins, have only been worked out on how they work in the last 10 years. And that big bag, you can see it pulling along, is a bag of molecules. So let's watch this in action replay. Here it is, walking along. It looks almost as if it's alive. That's a molecule. It's not alive. It's a molecule that's moving and being driven by energy, and it walks along a molecular road, pulling along a bag of molecules. How good is that? And once we understand how these work, we can design our own molecular machines at the molecular level. Okay, so I'm just looking at the time, and I might just scan along through here a little bit. I've just got to show you a little bit of work from Drew Berry. He works down in Melbourne, and he's one of the greatest animators in science visualization at the moment. And what you're going to see is DNA. You might not know too much about what DNA is, but it's a long molecule inside your body. But I want you to see how this molecule is curled up on itself, because it's an extremely long molecule, but it can't be curled up in a totally haphazard way. In a bacterium it is, but inside our cells, the DNA is an incredibly long molecule. So watch how it's curled up on itself into things called chromosomes. Chromosomes. And it's these chromosomes that are stored in the nucleus inside your cells. Okay, so watch this.
And it's jiggling and wiggling there because of the surrounding supermolecules, which are stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of this field of view is a single strand of DNA. This huge package of DNA is called a chromosome. And we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA, called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules. That name's not so important, but we're going to...
Remember those microtubules? So the lines you're seeing are those microtubules that the little molecules walk along?
If I focus on this red region, I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes. We have no idea, really, as to how it's achieving that movement. We've been studying this thing called the kinetic core for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. and you'll see that there's one little last bit that's still remaining red, and it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale.
There you are. So there's more of those motor proteins. You can see that this is all really complicated molecules interacting with each other like little molecular robots. How this works is now the subject of a lot of interest. Okay, let's have a bit of fun, because one of the things, we've only got 5 minutes, yeah? Oh, 10 minutes, good. Oh, I've been told 10 minutes. Okay, first of all, if you've done any chemistry at all, your teacher might have shown you the difference between graphite, which is what you get in the end of a pencil, and it's sheets of carbon atoms that can slide over one another. So that's why pencil is slippery, right? Another form of carbon is diamond. Diamond also contains exactly the same atoms, carbon atoms, but the atoms aren't in sheets. They're in a sort of a cage, and that's why diamond is really strong, but pencil lead is slippery. Now, here's the question. In your body, in the table on the screen at the moment, you can see all the percentage of your body, which is oxygen atoms, carbon atoms, etc. And so if, say, you weigh 50 kilograms, for example, 9 of those kilograms are carbon atoms. Okay? So I want you to hold that thought. 9 kilograms of carbon atoms in your body. Now, here's the horrific thing. If and when you die, if you get cremated, your body's burnt and all the molecules go off, and the carbon atoms usually go off as carbon dioxide molecules. Now, it's possible to collect that carbon dioxide, strip away the oxygen atoms, and perhaps you could produce diamond from the carbon atoms in your body. There's already a company that does this in, of all places, the United States. So you can turn, your pet dog into a diamond, and that's fine. Now, here's a sexist thing. Now, the girls in the audience might be thinking, oh, that'd be quite nice. I like diamonds. Now, the guys are probably thinking, what a useless thing to do with the carbon atoms. I can hear what you're thinking, guys. I would much prefer that carbon to be in the form of pencil lead, because it would be much more useful, wouldn't it, to have those carbon atoms in the form of pencil lead. And then, of course, if you came across, you wanted to remember a phone number or something like that, you could write it down. I mean, pencil lead's a useful form of carbon, whereas, diamond just scratches things. Now, you might think I'm joking, but once again, in the land of crazy ideas in the United States, there's a company that will do just this. It'll produce pencil lead from the carbon atoms in people who are cremated. So on the screen at the moment, you can see a pencil here, which says Joseph Wald, who lived from 1930 to 2003, and the pencil lead in there contains the carbon atoms in his body. What a great idea, isn't it? I mean, if your mother-in-law died, she could be more useful dead than she was when she was alive. What do you reckon? Eh? Oh dear, never mind. I haven't got time to talk to you about chemical systems, living chemical systems. What I will do is I'll zoom down now, because I've only got a few minutes left. Seriously now, I'm going to give you a really big idea. So if you've fallen asleep, now's the time to wake up, because I want you to think about this idea. Now, you know what a sand dune is. And sand dunes have a particular shape, don't they? In the desert, there are sand dunes that are shaped like a crescent, like that. And they move through the desert with the wind. And as the wind blows, some sand particles leave and new sand particles join the sand dune. And so sand dunes keep the same shape, but they can move for kilometers and kilometers. Now, think about that sand dune 100 kilometers away. Is it the same sand dune as it was earlier on? because every sand particle in that sand dune is different to the sand dune it was before. Okay, hold that idea. Now let's switch to you. You. Do me a favour now. Go with me on this. I literally want you to close your eyes. I want you to close your eyes and think of your earliest possible memory. What was your earliest possible memory? Maybe you were a child. I want you to try to remember where you were, the people around you, the colours. Is it a rich memory? Was it a birthday party? Okay, open your eyes. Think about that memory. Now here's the big idea. You think you were there when you had that memory. But you know what? You weren't. You are a bit like that sand dune. As you're going through life, you're taking in molecules, you're eating, drinking, taking in new molecules, and losing molecules. And so all of the molecules in your body are being exchanged and being replaced. Here's the big point. When you had that, if that memory was early enough, there wasn't a single molecule in your body when you had that memory that's in your body now. It's a bit like a car which is travelling along the road and bits are falling off and you're replacing it with new bits. The car still looks the same a long way down the travel, but no part in that car is the same as it was 200 kilometres before. So as you go through life, you're exchanging your molecules with the environment. You are not the person now that you were. So if you're not your molecules, what are you? Think about that. It's a really big idea. Thanks for listening. And I hope you've got some questions.
Unmute.
I got to unmute?
They unmute.
Oh, they unmute. Okay, right, good. They're still working out why they're made of molecules.
We have a question from Richmond High School.
Great.
Because matter cannot be created or destroyed, does that mean that when you're born and you grow, does that mean that the atoms and molecules that make up you have been recycled?
Absolutely. That's a beautiful question, and I think this is probably the first time that you've really thought about this. All the molecules in your body when you die are released to the environment, recycled into other people. And it's like you've got a big box of Lego and you build up a body out of Lego blocks, and then when you die, those Lego blocks go back into the pool and you produce a new person and everything else, and it's all totally recycled. We literally are at one with the universe because we're continually being produced, and then we go back into the pool of atoms and molecules. And as I said, as you move through your life, you're exchanging those molecules as you go. Thanks, that's a great question. Come on, the other schools, come on. You've got a question. No, there's stunned silence. Well, come on, let me ask you a question. I just gave you a question and said, what are you? If you're not your molecules, then what are you? What is the same about you now that you were when you were 3 or 4? What is the same if it's not your atoms and molecules?
Ask them to unmute to answer.
Oh, you've got to unmute to answer, do you? Yeah. Right.
Are we the environment that was around us as molecules were lost and you got new ones?
Yes. Okay. Sorry, keep going.
We are made of what was around us when our
that's true. I mean, the new atoms in your body now are the atoms that largely came from food that you've brought in and you've exchanged. And so you've been interacting with the environment, you've been breathing in molecules, breathing out molecules. Now, but what is the same between you now and you when you were 3? What is the same? Are you the same person? DNA! Hey, that's great. Well done, because the DNA is what defines you. But what's really interesting is that the DNA molecules are not the same DNA molecules that you had when you were really young. But what's happened was that if you can think about the DNA molecules when you were 3 as a necklace, and the order of the beads determines who you are when you're 3. Now imagine you keep on replacing the beads with the same ones, so your DNA now has the same order of beads. So you're still defined by your DNA, but they're just different beads. But the order is the same. So your DNA, the order, if you like, the order of letters is the same, but they're different letters. So I've been told to wrap up, and I want to thank you very much for listening and thinking about the molecular world. And if you're interested, this is a really exciting area to get into.